Hello everyone. So today we are going to read about metabolic interrelationship between major tissues during the starvation. So we'll talk about the metabolism of liver, that adipose tissue, the muscles and the brain, how they utilize various substrates like carbohydrate, protein and lipids in the starvation phase. So glucose is the fuel of choice for brain and muscles, but unfortunately the carbohydrate reserves of the body is so low that it cannot meet the energy requirement even for a day. So the fuel stores or the energy reserves of a 70 kg normal man are basically like triacylglycerol is like it is 1,35,000 calorie, and protein which is present in the muscle is like 24,000 calorie, and glycogen which is stored in muscles and liver it is 800 calorie. So the survival time of an individual on starvation is mostly dependent on the fat stores. So, and for this reason the obese individual can survive longer than the lean individual without consumption of the food during the starvation. So protein is basically the structural constituent and mostly present in the muscle. However, during starvation, protein can also meet the fuel demands of the body. So it is estimated that about one third of the body's protein can be utilized towards the energy needs without compromising the vital functions. So starvation is associated with the decrease in insulin level and increase in glucagon level. So the metabolic changes during the starvation, as discussed, to the major organ and tissue in this video, like in liver, muscles, brain and adipose tissue. First of all, let us talk about the liver. So in the starvation the glycogen reserves are utilized. So glycogen is converted to glucose, and eventually the glucose is converted to pyruvate, and pyruvate is converted to acetyl coenzyme, and this acetyl coenzyme can be converted into ketone bodies during the starvation. Also, this acetyl coenzyme enters the Krebs cycle for the generation of the energy. So liver in starvation, the carbohydrate metabolism in liver, which is an important function of the liver, is to act as blood glucose buffering organ. So the action of liver is to suit the metabolic needs of the body. So during starvation, increased gluconeogenesis and elevated glycogen degradation leads to the glucose maintenance in the blood. So mostly the glycogen degradation will lead to energy demands in the brain. So whereas in the lipid metabolism, the fatty acid oxidation is increased with the elevated synthesis of the ketone bodies in the lipid during the starvation. So this is due to the fact that the tricyclic acid cannot cope with the excess production of the acetyl coenzyme. Hence, the acetyl coenzyme is diverted to produce the ketone bodies during the starvation. And let us talk briefly about ketone bodies, which effectively serve as the fuel source for the peripheral tissue. The brain slowly adapts to itself to use the ketone bodies. So after 3 days, like after the 3 days of the fasting, about one third of the brain fuel demands are made by the ketone bodies, while after 40 days of the starvation, they contribute about 70% of the energy needs. So now let us discuss about the adipose tissue in the starvation. So there is increased amount of utilization of the fatty acid in the adipose tissue. So this fatty acid is eventually converted into acetyl coenzyme. This acetyl coenzyme is used in the Krebs cycle for the generation of the energy. So this fatty acid come from the degradation of the triacylglycerol, and triacylglycerol also participate in the formation of the glycerol. So this fatty acid which is degraded in the adipose tissue will be utilized in the liver. So this fatty acid which is degraded in the adipose tissue will be transported towards the liver. So this fatty acid is produced from the adipose tissue, transported to the liver, and this fatty acid will eventually be converted into acetyl coenzyme, and this acetyl coenzyme will be used as the generation of the ketone bodies, or can be diverted towards the Krebs cycle for the generation of the energy. Also, this fatty acid that is derived from the adipose tissue will enter in the muscles for the Krebs cycle metabolism. So what happens in the adipose tissue in the starvation? The carbohydrate metabolism, the glucose uptake and its metabolism are lower. Whereas in the lipid metabolism, there is degradation of the triacylglycerol the degradation of the triacylglycerol is elevated, and which is leading to the increased release of the fatty acid from the adipose tissue, which serves as the fuel sources for various tissues like brain. Brain is the exception here because the ketone bodies are utilized in the generation of the energy during the starvation. The glycerol liberated in the lipolysis serves as the precursor for the glucose synthesis by the liver. So the synthesis of the fatty acids and the triacylglycerol is totally stopped in the adipose tissue. So the synthesis of the fatty acid is totally stopped, but the triacylglycerol generates fatty acid, but this fatty acid is utilized in the generation of the acetyl coenzyme in the adipose tissue, or this adipose tissue is transported to the liver for the generation of the ketone bodies, or this fatty acid is transported to the muscles for the Krebs cycle metabolism. So the skeletal muscles in the starvation. What happens in the skeletal muscle in the starvation? So we have the fatty acid in the liver, and this fatty acid in the liver is transported to the muscles, and this fatty acid is converted into acetyl coenzyme, and this acetyl coenzyme participate in the Krebs cycle. There is also increased amount of degradation of the protein, which leads to generation of the amino acid. So we have glycerol and amino acid. So we have increased amount of glycerol and amino acid. So from this glycerol and amino acid, the gluconeogenesis occurs in the starvation, and from the gluconeogenesis, the pyruvate is formed, and this will eventually leads to the production of the acetyl coenzyme. So what happens in the carbohydrate metabolism in the skeletal muscle? So glucose uptake and its metabolism are very much decreased, and in the lipid metabolism, both fatty acid and the ketone bodies are utilized by the muscle as the fuel source. However, on prolonged starvation beyond 3 weeks, the muscle adapts to the exclusively utilized fatty acids. So this further increases the level of ketone bodies in the circulation. Now what happens to the protein metabolism? So during the early period of the starvation, muscles proteins are degraded to liberate the amino acid, which are effectively utilized by the liver for the glucose synthesis, that is known as the gluconeogenesis. On the prolonged starvation, however, protein breakdown is very much reduced, because I have already told that only 1/3 of the protein is utilized of the total protein, even if the person remains in the prolonged starvation. Now let us discuss about the brain. What happens in the brain during the starvation? So the major energy source for the brain is the ketone bodies. The ketone bodies in the brain are converted into the acetyl coenzyme, and the acetyl coenzyme participates in the Krebs cycle. Also, some amount of glucose that comes from the gluconeogenesis will participate in the brain energy, like the glucose is transported and it is converted to pyruvate, and pyruvate is converted to acetyl coenzyme, finally leading to the Krebs cycle metabolism, which will lead to the generation of the ATP. So as already stated, glucose is preferred fuel source by the brain. During the first 2 weeks after the starvation, the brain is mostly dependent on the glucose supplied by the liver gluconeogenesis. So this in turn is dependent on the amino acid released from the muscle protein degradation. So starvation beyond 3 weeks generally results in the marked increase in the plasma ketone bodies. So by this time, the brain has already adapted itself to depend on the ketone bodies for the energy needs. So so the basically in the liver there is the gluconeogenesis, also there is the production of the ketone bodies, whereas in the adipose tissue there is increased amount of triacylglycerol breakdown, leading to increased amount of the fatty acid, but this fatty acid will eventually used on the transported to the liver, and they utilized for the generation of the acetyl coenzyme. And in the liver, the fatty acid will be utilized for the generation of the acetyl coenzyme, which will be incorporated in the Krebs cycle. Also, there is increased amount of degradation of the protein, which will eventually liberate amino acid, and this amino acid is utilized for the gluconeogenesis. And in the brain, the preferred source is the glucose. However, during the prolonged starvation, the brain has already adapted itself with the ketone bodies for the generation of the energy. So I hope you understood this lecture. So this concludes our metabolic interrelationship among major tissues during the starvation. Also, this is the last video of the biochemistry metabolism. So I hope you understood this lecture. So this concludes our topic. Thank you.